In clinical oncology, identifying the exact somatic variant, a genetic mutation unique to a patient's tumor, determines which precision drugs will be effective. This specific biological signature dictates the entire treatment strategy. But sequencing machines cannot read a human genome linearly from start to finish. To process the DNA, they must shatter it into millions of tiny pieces, usually around 150 base pairs long, called reads. The immediate result is a massive, highly chaotic data file. Alongside actual genetic information, this file is crowded with machine errors, chemical typos, duplicated fragments, and artificial adapters from library preparation. The clinical risk hidden in this noise is significant. If a random machine error is misinterpreted as a real mutation, a patient may be prescribed an ineffective or even harmful chemotherapy. Because of that risk, raw sequencer data cannot be used for diagnosis. Before a doctor ever sees the results, every data point must survive a series of bioinformatic filters, the quality control pipeline. The process begins with the FASTQ file. This format stores every sequence read alongside a specific accuracy grade for every single letter. The Q score is the metric for base accuracy. The sequencer evaluates every letter, assigning a confidence value. Clinical labs enforce the strict Q30 standard, a 99.9% accuracy rate. Software trims the data, snipping away low-quality bases and adapter sequences, leaving only high-confidence information. This first checkpoint ensures that the subsequent analysis is built on a foundation of validated data points. Clean reads lack biological meaning until organized. The software uses a standardized reference genome, like GRCH38, as a master point of comparison. During alignment, algorithms map millions of short reads to their biological positions along the reference genome, organizing fragments into a BAM file. We evaluate this using the mapping rate, tracking the percentage of total reads that successfully matched a coordinate. We also measure the breadth of coverage. While mapping rate looks at the total data, breadth calculates what percentage of the specific target gene was actually reached by those matching reads. High mapping and breadth metrics provide the biological context necessary to begin searching for actual mutations. Now the software scans for variants, single-letter changes called SNVs, or small insertions, where the patient's DNA differs from the reference. To verify a mutation, we examine the vertical cross-section, known as read depth, which provides statistical evidence. For cancer testing, a 500x threshold means the sequencer checked that exact coordinate 500 times, providing a massive sample size to confirm findings. We then calculate the variant allele frequency, or VAF. This tracks what percentage of those 500 reads carry the mutation, helping to differentiate between true variants and the complex cellular mixture within a tumor. Combining depth with VAF thresholds filters out PCR duplication errors and random noise. Only true mutations are exported into a final, compact VCF file. The VCF file represents the final collection of verified findings, the result of compressing billions of data points into a few specific genetic signals. We have now narrowed an overwhelming volume of genetic data down to just a few verified variants. The next step is annotation. Bioinformaticians cross-reference these variants against global databases like ClinVar and GNOMAD to understand their impact. Population frequency is a key filter. If GNOMAD shows that a variant is present in more than 5% of the healthy population, it is categorized as a harmless natural variation rather than a cause of disease. These findings lead to the ACMG classification. Following strict international guidelines, every variant is placed on a 5-tier scale, from benign to pathogenic. This classification is the final result delivered to the oncologist. It provides the confirmation needed to determine if a mutation is a harmless trait or a disease-causing threat. In clinical sequencing, data quality is the basis for trust. By enforcing strict Q scores, demanding high read depths, and ensuring precise alignment, we convert a massive dataset into the specific evidence an oncologist needs to select a treatment.